Hello, my name is Rishabh Kapoor, and today I'll be presenting my research on the influence of nuclear to cytoplasmic ratios on transcriptional activation in Xenopus frog embryos, which I conducted this summer in the Skotheim lab. So, to begin, a defining feature of embryonic development in animals is that the early embryo is transcriptionally inert. In other words, all of the transcripts that are being translated in the embryo are coming from the maternal oocyte or egg cell. And so it follows that a key developmental transition occurs when the embryo degrades its maternal transcripts and begins transcribing its own genome. This transition is referred to as the maternal to zygotic transition, or the MZT. And since the 1980s, there's been a widely held hypothesis that the MZT is caused by an increase in the nuclear to cytoplasmic ratio. This means that as the embryo is rapidly dividing through the first several divisions of embryonic development, the ratio of the total DNA content in the embryo to the cytoplasmic content increases. And this increasing DNA content is thought to titrate out an inhibitor of transcription, thus allowing for the initiation of zygotic gene transcription. However, in recent years, a variety of competing models have emerged which posit that the MZT is in fact independent of the nuclear to cytoplasmic ratio. So to contribute to this debate, we asked the question, do decreases in embryonic ploidy, in other words lower nuclear to cytoplasmic ratios, delay transcriptional activation? To answer this question, we analyzed two hybrid embryos. The control condition was triploid, and the experimental condition was haploid. Both were hybrids constructed with one equivalent of the Xenopus tropicalis genome and some equivalents of the Xenopus laevis genome depending upon the condition. And if an increase in the nuclear to cytoplasmic ratio does indeed underlie the MZT, we would anticipate that the haploid embryos, the experimental embryos, would activate later than the triploid embryos because the haploid condition has a lower nuclear to cytoplasmic ratio than the triploid. So in order to make this comparison, it first becomes necessary to be able to rigorously define activation times. And this is what I spent most of the summer doing, is devising an algorithm that could detect the activation time. So the first step of the algorithm was to renormalize the RNA-seq transcripts per million data and then apply a filter to remove noisy fluctuations in this data. And so the key insight for this filter is that these expression levels should be either constant before the MZT or consistently increase after the MZT at this stage of embryonic development. And so the filter essentially, as we can see in the panel on the right, removes upward spikes, as we see on the top, or downward spikes, which deviate from the biologically expected behavior. And so having removed these noisy points, the next step was to fit a function to the remaining points. And the most logical function choice was a smoothing spline. So the smoothing spline allows us to produce a fitted curve without making any underlying assumptions about what the shape of that curve should be. It also allows us to fine-tune a parameter that dictates how tightly the curve fits to fluctuations in the data. And so this smoothing parameter, if sufficiently high, will allow us to produce curves as we see on the right, where we're not actually directly passing through every point, but rather we're producing a reasonable curve that could underlie the random variation in those points. And so having produced these spline fits, the final step was to then define a threshold expression level as the activation time. And so after scrolling through genes and kind of inspecting by eye, I settled on 20% of the maximum expression as my threshold. So for every gene, I defined the activation time as the first point where we saw at least 20% of the maximal expression level. So to give you a little bit more insight into how we selected the smoothing parameter, which dictates how tightly the spline fits to fluctuations in the data, Here is a plot of the interreplicate correlation coefficient versus the smoothing parameter. And our strategy was to pick a smoothing parameter that gave us a reasonably high correlation coefficient, but also was high enough or stiff enough that we were not overfitting to noise in the curves. And so we selected a smoothing parameter of about 0.40, which gave us a very high, close to the absolute maximum interreplicate correlation coefficient and reasonably stiff curves. It's also worth noting that if we compare the graph on the right, which shows us our correlation coefficients with the noise filter, versus the graph on the left, which is without the noise filter, the filter significantly improves our interreplicate correlation coefficients, indicating that it works. So now that we have rigorously defined our activation time algorithm, we could do downstream analysis, which revealed several positive results. So to begin with at the selected smoothing parameter of about 0.4, when we plotted the activation time in one replicate versus the other, we got a strong, significant positive correlation, which confirms to us that the algorithm and its detection of activation times is replicable and reproducible. More importantly, for our underlying experimental question, if we compare the activation time in the experimental condition, or the haploids, to the control condition, or the triploids, we see that on average, the experimental condition activates later. And so as you can see in this example curve in the middle, the pink curve corresponding to the experimental condition has a later activation time as designated by the pink vertical line than the control condition as designated by the blue vertical line. And indeed, this extends when we consider all 500 genes, as we can see in the cumulative density plot on the right. More than 80% of the experimental minus control activation times are positive, meaning that for most of the genes, the experimental condition is activating after the control condition. So to summarize the conclusions from the summer, we were able to devise a reproducible activation time detection algorithm that was robust to noise in the data. And when we applied this to our experimental and control condition, we saw that genes activate on average half a cell cycle later in haploid versus triploid embryos. So crucially, we can attribute this delay to the lower nuclear-to-cytoplasmic ratio in haploids, because the hybrids were specifically designed to have the same number of Xenopus tropicalis gene equivalents, so these differences cannot be attributed to differences in gene dosage. And so in aggregate, these findings lend support to the model that the MZT is caused by an increase in the nuclear-to-cytoplasmic ratio in Xenopus embryos. But we want to build on this. So next direction would be to compare the biological characteristics of early acting genes versus late acting genes. And as an example, we would be curious to see if late acting genes are indeed downstream of early acting genes in known developmental cascades in the Xenopus embryo. Thank you very much.